Greetings! Let's continue with our explication of how insects are good for you and actually have helped mankind and science in general. Today we're going to talk about one of the ways in which insects have shaped modern biology. And we're going to do this in 3 chunks. We're going to start with a brief introduction to Darwin and what he did. We'll then go on to discuss in a little bit of detail the fundamentals of genetics as established by Gregor Mendel. And then finally we'll get around to the truly relevant part of the lecture, which is to discuss how Thomas Hunt Morgan discovered the nature of mutation using another insect, in this case the fruit fly. So, we've had a tendency to think in this course about insects as unimportant nuisances, but history reveals a much richer and sometimes pivotal role for our friends with 6 legs. In fact, some of the most important cornerstones of modern biology were set in place with the help of the phylum Arthropoda. So today what we're going to focus on is how research on insects filled a critical hole in our knowledge base, the resolution of which led to this wonderful thing we call the modern synthesis. But in order to get there, we're going to return to the year 1859. This was a seminal year in which a book entitled On the Origin of Species by Means of Natural Selection was published. The author, I'm sure you already know, was Charles Darwin. He had actually been working on this book for 20 years. In fact, we like to joke, those among us who are professors and who endured the ravages of being an assistant professor, we like to joke that Darwin, were he working today, never would have gotten tenure because it took him over 20 years to publish his magnum opus. At any rate, he might not have published even in 1859, except he was about to get scooped. A much younger man, whose name was Alfred Russel Wallace, had happened upon much the same theory while recovering from the ravages of malaria in Indonesia. In fact, he was in what would probably be described as a drug-induced stupor when he came across the idea. So Wallace, having had this great idea, writes a letter to none other than Charles Darwin, the other proprietor of the same idea, with a short manuscript entitled On the Tendency of Varieties to Depart Indefinitely from the Original Type, which sounds, terrible. What the hell does that even mean? What it's talking about is the formation of new species, in essence. Darwin himself, when he got this letter from Wallace, realized that Wallace had basically just restated Darwin's theory of natural selection without using that term, that is to say natural selection, and in much shorter form. He was a bit panicked, and Darwin consulted his good friends Charles Lyell and Joseph Hooker, who convinced Darwin to jointly publish his 800-page book alongside Wallace's much shorter essay. So they did that, and the idea that species could change through the agency of natural selection was established. Now, what did Darwin in fact have in mind? And we recognize in saying this that yes, Wallace came across the same idea, but Darwin had worked out the mechanics in much greater detail. So his basic idea about how species change was fairly simple. His first premise is that, well, it's something that we would take for granted today, namely that there exists in nature variation. In order to verify this, you only have to look at the members of your own family. If you have siblings with the same biological parent, it will be pretty clear that you look different, except for identical twins, of course, even as you have basic similarities. So variation across a range of different traits, some of you have brown hair, some of you have blonde hair, some have brunette, even within the same family. You can find this kind of variation, and that's very normal. You wouldn't expect everybody to look precisely the same, even those of you who share the same parents. The second part of Darwin's argument was that the variation he observed, and that you can observe, was heritable. That is to say, it could be inherited. It can be passed down to new generations. Today, we would probably just say that these traits have a genetic basis. Thirdly, through copious studies, travels, and conversations with learned colleagues, this is what he was doing for those 20 years, Darwin believed that there were more offspring born than can be supported by the environment. In other words, there is in nature a chronic mismatch between the number of mouths you have to feed and the available resources. And this led to what was popularly called the struggle for existence, which is not a phrase coined by Darwin, but which is often used to describe this situation. That is to say, more organisms needing to be fed than food with which to feed them. The fourth element of Darwin's thesis was that organisms possessing favorable variation, which is to say best suited for the present environment, would leave more offspring because they had good genes, helping them to negotiate the environmental stresses and strains that they would be under. Finally, over long periods of time, which is to say geological time, millions or even billions of years, accumulation of small changes could lead to major alterations in the lineages of organisms. So this is the speciation part of the argument. Now, ever the reductionists, we sometimes contract Darwin's lengthy argument and the supporting evidence into the variation on life on Earth comes from descent with modification. Okay, so all those 5 arguments that I just gave you can be reduced to this phrase, descent with modification. And basically, we're talking about a family tree with each node or branch point being the part where new species are formed from a common ancestor. So this is a graphic that was actually drawn by Darwin and published in one of his books, showing the major acts of speciation for a particular lineage. And here you can see the kind of branches that I was alluding to, and the creation of new species from a common ancestor. One of the amazing things about which Darwin didn't know is that he had no idea how genetics and the passage of genes from one generation to the next worked. A second thing that he had no idea about is he had no clue where the variation upon which his theory depends came from. He knew it existed, but he didn't know where it came from. So he lacked 2 relatively key elements to buttress his theory. But he hypothesized that eventually both of these things, both how heredity works and where variation comes from, would be discovered. And when they were discovered, they would be supportive of his theory of natural selection. But this is one of the reasons why it took him so long to publish his book. He realized that these were major impediments in his argument. And without this information, he was very vulnerable to all sorts of criticisms. And so he postponed publication until, as we have pointed out, he was about to get scooped. Now, genetics and the mechanism of how heredity worked were actually worked out by a contemporary of Darwin, whose name, I'm sure you know, is Gregor Mendel. Mendel was an Austrian monk who had kind of a checkered history. He floundered as a farmer on his family farm, and eventually, because he preferred not to go into the military, which was one of the 2 major occupations for men at the time, he decided to enter a monastery, which is the other major occupational outlet. Mendel experienced several more failures, such as twice failing the exam to become a teacher. Even when he settled on performing biological experiments, his initial choice of test subjects, which were mice and bees, also proved to be disastrous. So up to that point, it was one failure after another. But eventually, he turned his attention to pea plants, and that turned out to be a very important and extremely fateful decision, because he was able to deduce the fundamental laws of genetics and become the father of genetics as a result. So simply stated, the laws that he was able to promulgate are as follows. First of all, the law of segregation, and secondly, the law of independent assortment. With these two laws, Mendel correctly identified the fact that genetic elements, later to be called genes, would obey these two fundamental laws, one of segregation, secondly of independent absorption. But unless you're a geneticist, that probably makes no sense to you, so let's take a more detailed look at how these things work. First, we need to appreciate the genius of Mendel's choice of pea plants. He focused on 7 traits for those pea plants that involved things like flower color and seed shape, seed size, and a few others. So you see illustrated here in graphic some of those traits. We cannot tell if these trait choices were fortuitous or whether he could tell that all 7 of these traits acted in a very reliable way in breeding studies. In all probability, he had some inkling that these 7 traits were special in that reliability, and for that reason he focused on them. But either way, this choice of pea plants and the 7 particular traits he decided to follow in his breeding studies made all the difference. The 7 traits selected by Mendel set him up for success because each trait, and here we get into some of the details that you need to know and understand, each trait, whether it was seed size or seed shape or flower color, each of those traits had 2 forms of each gene. We call these different forms alleles. We're talking about things like brown eyes versus blue eyes. It's the same trait, eye color, but different forms, brown or blue. And, moreover, for each of these 2 forms of the gene, the different alleles, the different forms, had a simple relationship of dominance to recessiveness. We'll talk more about it in a minute, but for this point, just note that had this not been the case, had there not been just 2 forms of the gene, had there not been a very simple and predictable relationship of dominance to recessiveness, Mendel might never have deduced the laws of genetics. So you'll get a sense as we go through this what it means to deduce something in a scientific sense. This is what Mendel did. What did he do? Well, Mendel began performing crosses among the different types of peas and focusing on the 7 characteristics that I've already mentioned. One of the things that he learned very quickly is that when parents in a cross differed in a trait, so if we're talking about seed shape, he'd be crossing one parent having a smooth seed by a second parent that had a wrinkled seed. If he did something like that, and this applied to all 7 traits, the offspring would show one trait or another, but not a mixture of both. Now this was very important because at the time the reigning theory of inheritance was the so-called blending theory. The idea was that if 2 parents differed in a trait, when you bred one parent to the other, you would get in the offspring a condition that was sort of halfway between each parent. For instance, if you bred a black cat to a white cat, according to the blending theory, the offspring should all be gray, in other words, halfway between the trait shown by the 2 parents. And what Mendel did, what Mendel added to the argument, was to show that blending was wrong. because in Mendel's experiments, when two parents differed in a trait, the offspring showed one trait or another, but not a mixture of both. Mendel also provided some insight into the other mechanics of heredity. His law of segregation, which we shall look at in a moment, helped us to understand that each parent, male and female, contributed equal amounts of genetic material to the offspring. That understanding grew from his so-called law of segregation. So up to that point, it was sort of a matter of debate as to whether males or females contributed more to the offspring, or whether they both contributed equally, or whether some contributed sometimes, not others. It was just sort of all kind of up in the air. Finally, Mendel demonstrated that the genes for different traits behave independently from one another. In other words, the gene for seed color had no dependence on the gene for flower color, and vice versa. And this became known as the law of independent assortment. So the question is, how did Mendel do all this? How was he able to deduce these very fundamental laws of genetics? Well, first he looked at very simple patterns that occurred in the crosses that he was making. And he was looking for things such as dominance and recessiveness of traits. Mendel had learned that dominant genes were so called because when a dominant gene or form of a gene was present, it would mask the appearance of a recessive gene. You also know this from your own life. The gene for brown hair in humans is dominant to the form of the gene, same gene, for blonde hair. So if we represent hair, and brown hair in particular with a capital B, and blonde with a small b, we can easily demonstrate this. Remember that both parents contribute equally to the offspring. Therefore, when both parents contribute a big B, then the baby will be big B in its genetic constitution, and it will have brown hair because brown is dominant. Big B is dominant. However, if one parent gives the baby a big B and the other a little b, then the baby genetically will be big B, little b, and will also have brown hair because big B is dominant to little b. That's what it means to mask a trait. Now, because the gene for blonde hair, which we've designated little b, is recessive, and its appearance will be masked by big B, the only way you can get a blonde baby is if both parents contribute a little b to their offspring. If both parents contribute a small b to the baby, then the baby will be little b, and will have blonde hair. So you have to have two copies of little b in order to have blonde hair, and this is one reason why blonde hair is relatively rare. You need two copies of that gene because little b is is recessive to big B. Before we go on now, let's take a moment to introduce a few more terms. When we refer to the actual genes that an organism has, we're talking about its genotype. So there are three genotypes in the human hair example, which were big B, big B, little b, and finally little b, little b. However, when we're talking about what an organism looks like, we're referring to its phenotype. From the example I just used, there are only two phenotypes because both big B and big B, little b will result in brown hair, while only the little b person will have blonde hair. Another set of terms we need to introduce are homozygous and heterozygous. Homozygous means that an organism has two copies of the same gene, that is to say big B, or little b, little b. Homozygous means it has only one form of that gene and it has two copies of it. In contrast, A heterozygous individual has mixed genes, or one copy of both genes, or big B, little b is its genotype. We can also mix terms like homozygous dominant, which means big B, or homozygous recessive, which means little b, little b. All right, let's return to Mendel. Mendel's test subject, the pea plant, was operating with the same system. That is to say, two forms of the gene, simple dominance and recessiveness of the two forms. What I said about heterozygotes, homozygotes, also germane to this discussion, and I'll probably use those terms as we go. Mendel was able to deduce what was going on by noticing the resurgence of particular patterns in the offspring from different crosses. He noticed two general patterns of inheritance, and they're listed for you on the left side of the slide. Sometimes in his crosses, he got 3 dominant phenotypes for every 1 recessive phenotype. And other times, he got all dominant phenotypes in his offspring. Here's how Mendel figured it out. Take any one of the 7 traits that he worked with, although you could do it with all of them if you wanted to. This particular example involves the texture of the surface of the pea's seeds. And there are 2 states in which they can exist. They can be either smooth or they can be wrinkled. So there are 2 phenotypes, smooth or wrinkled. Here we will represent traits with a big S for smooth, or with a little s to indicate wrinkled. In this experiment, Mendel was working with what we call true breeding types, which means that if you cross a smooth-looking parent with another smooth-looking parent, you will always get 100% smooth-looking offspring. Similarly, if you cross wrinkled parents by wrinkled parents, you will always get wrinkled offspring. So another way of expressing this idea, and that of true breeding, is that all of the parents were homozygous. Okay, if that doesn't make sense to you, go back and review the definition of homozygous and heterozygous. With this information, you should now be able to understand the cross that Mendel did. Mendel crossed one true breeding homozygous dominant parent, which would be S, big S, with a true breeding homozygous recessive parent, which would be little s. So the question is, if that's the cross, you have one homozygous dominant, one homozygous recessive parent, what would the outcome of that cross be? And the answer is, all of the offspring would be heterozygous. They would all be big S, little s, and phenotypically they would all show the dominant trait, which is to say they would be smooth. The easiest way to understand this, if the previous slide didn't make any sense to you, is to do what we call a Punnett square, which you see illustrated here. What we do is we represent the genes of the first parent along the top of the table and the genes of the second parent along the side. We're going to stipulate that parent 1 is a true breeding smooth parent. So you already know what genes it has. It will be big S, and it will only be able to give the big S allele to its potential offspring. Now we're going to indicate the genes, the alleles produced by the second parent, which is a little s individual. And in this case, because it's true breeding, just like the other parent, it can only produce the little s allele, and therefore that's all it can contribute to its offspring. Now what the Punnett square allows you to do is to actually do the cross. You write in what each parent is going to be contributing to each offspring, with each box representing a single offspring, and you can then visualize both the genotype and figure out the phenotype once you've done that. And what you see when you do this cross using the Punnett square is that each of the offspring produced will have one copy of the big S allele from parent 1 and one copy of the little s allele from parent 2. They're both true breeding, so that's all they can produce. And what that means is that all 4 of the potential offspring from this cross are going to be heterozygotes. They're going to have one copy of each allele, and because big S is dominant to little s, 100% of the offspring will be smooth. And this explains one of the patterns that Mendel saw. This example is one of the 2 patterns that Mendel found over and over again. And when he studied crosses made by true breeding parents for all of these 7 traits, he always got 100% dominant phenotypes in the offspring. The only way this can happen is if one parent is homozygous dominant and the other is homozygous recessive. This will give you all heterozygotes in the offspring, all showing the dominant trait. So this explains the first Mendelian pattern. But if we now take parents that are not true breeding, you will get a different pattern. So just to move things along, let's stipulate that if you take 2 non-breeding parents for the seed texture trait, what you are crossing would have the following potential genotypes. You could have all the offspring showing big S, and therefore be smooth. You could have the offspring being heterozygotes, big S, little s. There are actually 2 ways to do this. You'd have twice as many of those as the others, which we'll see in a minute. But because big S is dominant to little s, these 2 will be smooth. And finally, you could have the little s, or homozygous recessive genotype, and these, finally, would be wrinkled. So, we have 3 potential genotypes from this cross, big S, little s, and little s, but 2 potential phenotypes, smooth or wrinkled. So you should be able to look at those genotypes and tell me which ones are going to be which phenotype and why. Again, let's set up a Punnett square. Again, we will represent the genes of one parent, parent 1, along the top, and the second parent down the side. We've already stipulated these are not true breeding parents. So that the implication of that is that they're both heterozygotes. So both are going to have one copy of the big S and one copy of the little s allele. And that means that each of the 2 parents can potentially contribute one of 2 different alleles to their offspring. Each can potentially contribute either the big S or either the little s. And if you do the Punnett square and indicate the genotype of each offspring in the box, you can first of all see that yes, the 3 genotypes we previously discussed are possibilities for these offspring. You can also see that there are 2 ways to get the heterozygote, so they're going to be twice as abundant as either of the homozygous forms. And you can see that there's only one way to get the wrinkled form, having both copies of the recessive allele. So this is really exciting. Why is it exciting? Well, because this explains the 3-to-1 ratio of phenotypes that Mendel routinely saw. When he crossed heterozygotes, he got 3 potential genotypes, but 2 potential phenotypes, and in a 3-to-1 ratio. And his breeding studies allowed him to explain this result. So now we know how Mendel deduced the laws of inheritance, a highly experimental and mathematical demonstration of how nature worked. You too should be able to explain how Mendel arrived at his conclusions. Maybe you can appreciate the difficulty of operating in the dark without key pieces of information, like what a gene is. They still didn't even have that term during Mendel's time. So this is considered sort of a hallmark of good science, quantitative science, experimental science, and deductive science. Sadly, things did not work out that well for Mendel, despite his ability to explain and unravel one of the key mysteries holding Darwin back. Mendel, at one point, sent a copy of his work. He had written up many of his experiments in a manuscript form, and he sent a copy of it to Darwin, who apparently never bothered to read it. Darwin never responded to Mendel's submission, nor did Darwin discuss it in any of his letters. It wouldn't be until 1900 when three scientists, de Vries, Correns, and Schermarck, simultaneously discovered Mendel's work and understood its importance and relevance to the fledgling field of genetics, that Mendel was finally given his due as having identified the mechanism of inheritance. But neither Mendel's work nor anyone else's could explain where that variation came from. And without that information, it was still not possible to explain speciation. There were lots of ideas floating around at the time about the mechanisms of speciation, or the creation of new species. There was, for instance, the Saltationist school, which proposed that new types of species came about by big jumps, not as proposed by Darwin, which would be by the accumulation of small changes over large periods of time. The Saltationists thought that new species would appear periodically and very rapidly, that you wouldn't have millions of years of change occurring to produce these new species, but rather that, in a very short period of time, big genetic changes would take place. Then there was the biometric school, which argued that variation in nature was continuous from one species to the next, with an abundance of intermediate forms. And that would be true in some cases. We can see a sort of progressive gradation of one type to the next, and lots of intermediate forms in between endpoint species. Finally, there was a theory of mutation that was advanced by de Vries, one of the previously mentioned co-discoverers of Mendel's work. And as it turns out, the theory of mutations was wrong, but was still a significant step forward for the time. The idea here was that the genetic material referred to in that time as pangenes underwent alterations called mutations. So at this time, turn of the century, 1900, the genetic particle that was transmitted from generation to generation was called a pangene, not a gene, but a pangene. And it was believed that the pangenes from time to time underwent alterations within the individual, although the mechanism for producing that alteration was not specified. It turns out that, at least in a descriptive sense, this was actually true. But the part that they got wrong was that they believed that pangenes were genetic material that were produced, thrown off by all of the body parts, and that the pangenes would travel to the gonads, where they would be taken up by sex cells and then passed on to the next generation. So we now know that that's definitely not true, but at least it was sort of a step in the right direction. Somehow, there needed to be an experimental resolution of which idea was correct. And here, as was the case with Mendel, the critical element was finding the right test organism. This is a theme that actually plays out through much of biology. As we've already learned, Mendel was either extremely lucky or incredibly astute in choosing pea plants, because he was able to work with an organism that had easy-to-follow traits that showed simple dominance and recessiveness. In truth, genetics, like just about everything else, is often much more complicated than that. And when you're trying to work out the fundamentals, as opposed to elaborating upon established principles, it's absolutely critical to have a cooperative, by which we mean simple, test organism to help you out. And help was on the way. Here, at last, we get to why insects are relevant to this quest. In the laboratory of a scientist named Thomas Hunt Morgan, he's a man who had done considerable work with the aforementioned de Vries, and was also kind of a mutationist, but had a much different understanding of the importance of mutation and how it worked. In fact, Morgan, although also a mutationist, had vivid arguments with De Vries about the theory, and were not really on the same page for much of it. Morgan benefited from his insistence that we needed to learn about mutation and its role in heredity by using experimental studies. And Morgan wanted to show that the production of mutations could, in fact, produce new species. In order to do this, he was making lots of studies using Drosophila melanogaster, otherwise known as the fruit fly. Morgan began working with fruit flies and subjecting them to radiation, the source of many a good sci-fi tale, in order to induce mutations. It was said that part of Morgan's success was due to his uncanny ability to pick out fruit flies from a swarm of individuals that looked different, that had some sort of change, which we now call mutation. With his super eyesight, Morgan found the so-called white-eyed male in 1910. He then, like Mendel, began doing breeding experiments. When he bred the white-eyed male back to a red-eyed, that is to say, normal sister, he found that all the resulting offspring had red eyes. But if he selected from the offspring of the white-eyed male and the red-eyed females a red-eyed female and a red-eyed male, he got a ratio of, wait for it, 3-to-1 red to white eyes in the second generation. So far, it sounds like a cross between two heterozygotes, right? Well, nope, that's not right. Here's a graphic of Morgan's experiment. And now we need to do a little more backpedaling to explain a weird thing that is going on. It's called sex-linked inheritance. Like humans, female fruit flies have 2 X chromosomes, and male fruit flies have an X and a Y chromosome. So if a particular gene, and this is getting to the critical part, is found only on the X chromosome, it means that female fruit flies will have 2 copies of the gene, while the male fruit fly will have only 1 copy of the gene. And if the gene has 2 forms of the gene, or alleles, which, like Mendel's pea plants, show simple dominance and recessiveness, then a female fruit fly with 2 copies of the gene could have 3 potential genotypes. We'll use R's in this example. The genotypes would be big R, secondly the heterozygote, or big R, and finally the homozygous recessive form, little r. And, just like Mendel's experiments, there would be 2 potential phenotypes, namely red eyes and white eyes. That's the female. However, a male fruit fly with only 1 copy of the X chromosome would have only 1 copy of the gene for eye color on the X chromosome. So there would be 2 possible genotypes, each one having a single allele. One genotype would be big R, the other potential genotype would be little r, and there would be 2 potential phenotypes, red or white-eyed. So importantly, because the male has only 1 copy of the gene, it will show the recessive phenotype with only 1 copy of the gene. You remember in Mendel's experiment, in order to show the recessive form, it would have to have 2 copies of the recessive allele. Well, not so in male fruit flies, because it only has 1 copy of all of the genes on the X chromosome, because it only has 1 X chromosome. So in that case, it can show the recessive trait when there's only 1 copy of the gene. This is called sex-linked inheritance. Let me explain this graphic, which is pretty complex. At the very top, we see a normal red-eyed female with 2 X chromosomes. The X chromosomes are off to the left there. You see the red-eyed female. And she's going to be, we're going to stipulate that she's big R, big R. Now she's going to mate to a mutant male that has white eyes, so you see the white-eyed male there at the right. And of course he's only got one copy of the X chromosome, which is painted green in this case. So the X chromosome is a little cigar-like thing that appears at the top, and then under that you have a copy of his Y chromosome, which has a different shape. But it does not have the gene because it's a Y chromosome, not an X chromosome. So the gene for eye color is not on the Y chromosome. And then below that you have a Punnett square, which shows how a cross between a normal female, which they've abbreviated in this case, instead of having two entries for the normal female along the side, you only have one entry because she's only making the big R allele, so she can only produce big R offspring, so they've simply abbreviated it to one possibility, which is correct, but probably not as helpful as having both in there just for completion. And along the top you see the white-eyed male. He's got one X chromosome that's carrying a mutant gene, or little r, and then he can also give the Y chromosome, which appears in his second box, and we need to know that information so that we know what the sex of the offspring is, since that will be important in determining whether or not the recessive trait will show. So if you do the cross in the same way that we have done before, you will find out that all of the female offspring, which appear at the left, having two copies of the X chromosome, all of the female offspring are going to be normal. They're all going to have two copies of the big R gene. On the other hand, the male offspring are going to get one copy of the X chromosome from their mother, and they are therefore going to be big R. And even though they only have one copy of the big R gene, they're going to be red-eyed because that's all it takes. So all of the offspring in this cross will be red-eyed, regardless of whether they're male or female. Okay, in a second cross with a red-eyed female carrier from the first cross, so she will be big R, little r, and she's going to be crossed with a normal red-eyed male, but because he's male and only has one copy of the X chromosome, he's going to be big R. And when you do this cross, things start to get interesting. The female parent in the second cross, we've already designated, is a heterozygote. So she can contribute either a big R or a little r to her progeny. And her genes are along the side, as we've done consistently through these crosses. In contrast, the male parent has only the big R gene. So that's all he can give to his progeny. And we're going to track the X chromosomes as well as the Y chromosomes so that we can determine sex and thus the phenotype of the progeny. So if we do the cross here, half of the female progeny, and now the female is going to have two entries because she can contribute two different alleles. Half of the female progeny will have two copies of the big R allele and will therefore be normal. The other half of the female progeny will be carriers for the little r gene and will be heterozygotes. All of the females will have red eyes. But among the males, half of the progeny will have gotten a normal big R gene from their mother and will have red eyes, and the other half will have gotten the little r gene from their mother. And because all the progeny are male, they will express the trait, in this case, white eyes. And the results will be in a 3-to-1 ratio of red-eyed individuals that are going to be mixed sex to white eyes in the second generation, and those will all be males. Okay, now this is complicated, I know that, so I would go back through it a couple of times and look at the graphic and correlate it with what I'm saying, because you will be asked a question or two about this. All right, Morgan's discovery of sex-linked inheritance opened up other avenues of investigation. Another trait which behaves the same is body color. where the options here are either a gray body type or a yellow body type, with the yellow being the mutant form. The mutant eye color gene and the mutant body color gene were always found together, which means that they were not segregating independently, as Mendel's second law stated. It turns out that this is because the two genes were found on the same chromosome, which is the X chromosome, and they were found very close together. And because of their proximity, that is to say, because of how close they were together on that same chromosome, no crossing over occurs between those two chromosomes, which would lead to their independent segregation. Now, this is not something that we've talked about very much in this class, so I'm not really going to expect you to know the mechanics of crossing over, but simply know that this is one case in which Mendel's basic laws do not apply when genes are very close together on the same chromosome, as is the case with these two mutant genes in fruit flies. They don't necessarily segregate independently. But thanks to Morgan's work, mutation was now understood to be the source of genetic variation in nature. In other words, departures from the standard, the normal type, whether it's a wrinkled seed or a white eye in fruit flies, those kinds of variation provide the substance upon which natural selection, discussed by Darwin, works. Morgan induced genetic changes, which is to say changes in the structure of DNA, using X-rays. And the use of the X-rays functionally just disrupted DNA and made mistakes in other genetic processes take place, such that the materials available for producing offspring themselves were changed. Mendel's work showed how Darwin's ideas about genetic variation were related to Mendel's laws, and both Darwinism and Mendel were compatible. Further, Morgan showed that mutation did not result in speciation in a single step. So the mutationists were wrong in that suggestion, but if you look at mutation as a process that over large periods of time can dramatically alter the genetics of a species, then Darwin's view appears to be correct. That is, speciation is the cumulative effect of extensive adaptive radiation driven by natural selection. One more monumental discovery was needed, and that came in the form of Watson and Crick, who discovered the three-dimensional structure of DNA, and this functionally tied all of the loose strands together into a coherent model, which we call the modern synthesis. The model of Watson and Crick set the platform for understanding how DNA and consequently heredity functioned, how information was precisely copied and packed into sex cells, how disruptions in the sequence of DNA could result in mutation, how mutation could either result in a functional alternative gene or a complete disaster. With Darwin's focus on heritable variation, Mendel's work on how traits were determined from genes, Hunt's work on mutation as the source of variation, and Watson and Crick's work on DNA structure, the picture was, for the first time, complete. As is true of all good theories, the modern synthesis provided great explanatory power and predictive power, which has driven the course of biological research ever since. Among the big ideas explained or correctly predicted, we have the following. We now have a mechanism for understanding why there is so much biodiversity on our planet. We now understand both how it is possible for you to look like your parents or not look like your parents. We understand that selection works at the level of genes, but within populations, not at the organismal level, as Darwin proposed. We have a better understanding of how new species can be formed from accumulated changes and by other mechanisms besides what Darwin hypothesized as well. Among these other explanations, we have, for instance, genetic drift. In addition, a variety of reproductive isolating mechanisms not known to Darwin, as well as the interaction of the genome with viruses and other contributors of DNAs to their cells. Is it right to attribute all of this knowledge to the fruit fly? Well, maybe not explicitly. But even Darwin's work depended heavily on examples that insects provided to biodiversity. The information provided by Hunt was critical in securing the modern synthesis, and with it, clearly benefited from the insect model that he developed. Could our understanding of mutation have arisen from some other test organism? Yes, it probably could have. But insects offered some huge advantages, like a short generation time, large groups of progeny produced, ease of handling and cultivation, and susceptibility to induction of mutation with experimental methods, and offspring whose mutations could be distinguished by their color. In genetic research, the challenge has always been to find the right organism. There is no doubt that the use of the fruit fly made it easier for Morgan to figure out how mutations worked. It could have happened with another organism, but it certainly would have taken longer. So in that sense, we are very lucky that the fruit fly was there to speed our understanding. All right, next time we'll look at another way in which lowly, apparently non-consequential insects have impacted the very foundations of science itself. See you then.